Ever wonder how scientists can give a plant a brand new ability? Like making it disease resistant or even letting it produce its own vitamins? Well, today we're basically opening up the instruction manual. Let's dive into this 6-step guide on how to create a genetically modified plant.
So here's our roadmap. We're going to walk through a 6-step recipe that takes us from identifying a big problem all the way to growing a brand new plant that's actually designed to solve it. It's a journey from an idea to a living, breathing solution.
Every great project, every recipe, it always starts with a goal. And in genetic engineering, that goal is usually about tackling some of humanity's biggest challenges. What's so important to remember is that this whole process doesn't start with some cool new tech. It always starts with a purpose.
So what kind of challenge are we talking about here? Well, how about this number? Over 2 billion people. That's more than a quarter of everyone on the entire planet. And this isn't about not having enough calories to feel full. It's what scientists call hidden hunger. It's when the food you eat just doesn't have the critical micronutrients our bodies desperately need to thrive. This can lead to all sorts of problems, like weakened immune systems and developmental issues, and it really hits women and children the hardest.
So, here's the big idea. What if we could take a food that people eat every day, like rice, and turn it into a source for those missing vitamins? What if we could give that plant a new instruction in its DNA that says, hey, start making vitamin A? That's the goal. That's the superpower we're trying to create.
Okay, so if we're going to change a plant's instructions, we first have to learn its language. And the language of all life is written in DNA. So everything we're about to talk about, it all builds on our ability to read and understand that fundamental code. The wild thing is just how simple and at the same time how incredibly complex this is. Every living thing you can think of, from a tiny bacterium to a massive blue whale, is built using an alphabet of just 4 letters: A, T, C, and G. The specific order of those letters is what forms the genetic code, a unique blueprint for every single organism. So how does the cell actually read this blueprint?
Well, it follows a fundamental process that scientists call the central dogma. The DNA code, which is the master copy, gets transcribed into a temporary message, a molecule called RNA. Then that RNA message is translated to build a specific protein, and proteins are the little machines that do all the work in the cell. You can think of this table as the universal dictionary for life. The cell reads that RNA message in 3-letter words called codons. Each one of those words points to a specific amino acid, which are the building blocks of proteins. And the most amazing part? This code is universal. It means scientists can read a gene from any organism and know exactly what protein it's going to make. Okay, so we can read the blueprint. We know the exact word we want to add to the instructions. But how do you actually go in and edit the text? For that, you need a very special molecular toolkit. The early days of genetic modification they were a bit clumsy, kind of like using blunt scissors in the dark. You could get a new gene in there, but you had almost no control over where it ended up. But modern tools, especially CRISPR-Cas9, were a total game changer. Think of it like a biological find and replace function. It lets scientists make incredibly precise edits at an exact spot in the genome. So how does this magic scalpel actually work? Well, you can break it down into 3 simple steps. First, a molecule called a guide RNA acts like a GPS, homing it on the exact DNA sequence we want to change. Second, an enzyme called Cas9, those are the molecular scissors, is brought to that exact location and makes a clean cut. And third, and this is the really clever part, the cell's own natural repair kit kicks in to fix the break. And while it's doing that, we can trick it into stitching in our new piece of DNA. All right, we have our new gene and we have our editing tool. But how do we actually get it past the plant cell's defenses and into the nucleus where all the DNA is kept? Well, for this, scientists looked to nature and found an expert. I want you to meet Agrobacterium tumefaciens. This common soil bacterium is, no joke, a natural master of genetic engineering. It's been transferring its own DNA into plants for millions of years. In the wild, this bacterium causes something called crown gall disease. It literally injects a piece of its own DNA into a plant cell, hijacking it. This forces the plant cell to start producing food for the bacterium and to just multiply out of control, forming a tumor. It's a little sinister, sure, but it's also incredibly sophisticated. So, scientists pulled off this brilliant move. They decided to hack the hacker. First, they disarmed the bacterium by taking out its tumor-causing genes. Then they loaded the good gene they wanted to add right into that same spot. So now, when this engineered bacterium infects a plant cell, it does what it's always done. It injects that piece of DNA. But instead of causing a tumor, it delivers our helpful new gene right where we want it.
So we've done it. We've successfully edited a single plant cell. I mean, that's an incredible achievement. But one microscopic cell isn't a plant. So how do you go from that one cell to a full-grown plant that you can actually put in a field? Well, the answer lies in a property that is pure biological magic. And that magical property is called totipotency. See, unlike most animal cells, a single plant cell has this amazing, hidden ability to grow into an entire, complete organism. It still has the instructions to make roots and a stem and leaves, everything. It just needs the right chemical signals to get started. The whole process is called tissue culture. First, that one modified cell is placed in a nutrient-rich jelly. Then, with the help of some plant hormones, it's encouraged to divide into a little clump of cells called a callus. Then, by changing the hormone cocktail, scientists can tell that callus, okay, time to start growing some roots. Now make a little shoot. And eventually, you have a tiny plantlet that you can move to soil where it grows into a mature plant that carries our new gene in every single one of its cells.
So we've followed the entire recipe. We started with a problem, we read the blueprint, we used our tools, we made the delivery, and we grew the final product. So what does this actually look like in the real world? The applications are already pretty staggering, and this isn't science fiction. We're talking about rice with more iron to fight anemia, citrus trees that can resist diseases that are threatening to wipe out the entire industry, crops that can actually survive a drought, soybeans that produce healthier oils, even decaffeinated coffee plants. Every single one of these started with the same basic recipe we just walked through.
So when you take a step back, what we've just explored isn't just a cool lab trick. It's a precise, powerful, and repeatable process. We have followed a recipe that lets us fundamentally and very deliberately rewrite the genetic rulebook for a living thing, all to solve a specific problem.
This technology gives us an unprecedented ability to shape the living world around us, to address disease and hunger and huge environmental challenges in ways we could only dream of before. So the question isn't just how we do it anymore. The real question is, now that we can literally edit the blueprint of life, what should we write next?